Ready to dive into the world of genomics. We've got a fascinating deep dive for you today.
Absolutely. We're dissecting a brand new article just out today, in fact, from Biology Notes Online.
September 18th, 2024. Talk about hot off the press. And it's all about high-throughput sequencing, or HTS.
HTS is really at the forefront of genomics research right now. It's changing the game in so many ways.
So for our listeners who might be new to this, let's start with the basics. What exactly is high-throughput sequencing?
Well, imagine trying to read an entire library, every single book, all at once.
Sounds impossible.
It would be with older methods. But that's essentially what HTS allows us to do with DNA and RNA. Analyze massive amounts of genetic information quickly and affordably.
So speed and efficiency are key here.
Absolutely.
Yeah.
It's a complete game changer compared to older technologies like, say, Sanger sequencing, which could only handle one DNA fragment at a time.
Yeah.
Incredibly time-consuming and expensive.
Okay, I'm starting to see why everyone's so excited about HTS. But how does it actually work, this massively parallel sequencing idea? It sounds pretty complex.
It is, but let me try to break it down. Imagine, instead of reading one book at a time, we have millions of people each reading different snippets simultaneously. That's kind of what HTS does with DNA or RNA. It breaks it down into smaller fragments and sequences them all at the same time, giving us this incredibly detailed view of the entire genetic code.
So it's like having this huge spotlight illuminating the entire genetic landscape.
Precisely. And there are different generations of HTS technologies, each with its own approach. Second generation HTS, for example, often uses a method called sequencing by synthesis. We're essentially watching DNA strands being built base by base. Platforms like Illumina use fluorescently labeled nucleotides, and we can actually decipher the sequence by capturing the light emitted as each base is added.
Wow, that's amazing.
It is pretty incredible. And then there's third-generation sequencing, which can sequence single molecules directly in real time.
Okay, now that's just straight out of Star Trek. Tell me more about that.
So technologies like PacBio and Oxford Nanopore can actually detect changes in electrical current or light as the DNA molecule passes through a tiny pore.
A tiny pore.
Yeah, it's pretty wild. And those changes tell us the sequence of the DNA.
That is seriously mind-blowing. But I think we need to take a step back and walk through the actual steps involved in HTS, because I'm sure our listeners are eager to know how this all comes together.
Absolutely. So it all starts with extracting the DNA or RNA from a sample, which could be anything from a blood sample to a tissue biopsy to even like an environmental swab.
So first, we need that raw genetic material.
Right. And then those long strands of DNA or RNA get broken down into smaller, more manageable fragments. Think of it like cutting up a massive document into paragraphs.
Okay, we're making those bite-sized pieces of information. What's next?
We attach these special adapters to the fragments. They're like unique barcodes that allow us to track each fragment during sequencing. Once we have our barcoded fragments, we create what's called a library.
A library of DNA.
Exactly. It's basically a carefully curated collection of all the DNA or RNA pieces that we'll be sequencing.
So organization is key here.
It's all about prep work at this stage. And this is where things might differ a bit depending on the specific HTS platform. With second-generation platforms like Illumina, we typically amplify the library, making many copies of each fragment to boost the signal.
Makes sense. And then?
And then finally, we load this meticulously prepared library onto the sequencing platform and let the magic happen.
It sounds like setting up a ridiculously complex domino chain, but one that can unlock this incredible wave of information about, well, life itself.
Exactly. And the discoveries we're making because of HTS are just mind-blowing, and the applications are just exploding. It's really incredible what we're learning about everything from healthcare to agriculture to, even forensics.
It's amazing to think about. And our article from Biology Notes Online actually lays out some of those applications in detail.
Yeah, it's a really great overview.
But before we get to those applications, I think we need to talk about the stars of the show, the different HTS technologies themselves.
Oh, yeah, absolutely.
Because, there's not just one type of HTS, right? Our article highlights 4 main players, and they've even given them these catchy little headlines.
Like a genomics magazine, I like it.
Exactly. So first up, we have Illumina, which they've dubbed the workhorse.
Yeah, Illumina is kind of the tried and true workhorse of the HTS world. It's known for its affordability, it can handle huge volumes of DNA at once, and the protocols are very well established. So yeah, workhorse is definitely a good way to describe it.
Reliable, efficient. But I imagine every technology has its pros and cons. What's the trade-off with Illumina?
Well, one thing is that Illumina tends to produce shorter reads compared to some of the other technologies. It's still incredibly accurate, but those shorter reads can make it a bit trickier to assemble really complex genomes. Like imagine trying to put together a puzzle, but all the pieces are really, really small. You might eventually get the overall picture, but some of the finer details might be harder to figure out.
So shorter reads, but still incredibly powerful for a wide range of applications, I imagine.
Oh, absolutely. Illumina has really revolutionized so many areas of genomics.
Okay, on to our next contender. We have Thermo Fisher's Ion Torrent, aptly named the Speed Demon.
As you might guess, Ion Torrent is known for its speed. It can churn out sequencing data incredibly fast.
So when you need those answers quickly.
Ion Torrent is your go-to.
Exactly.
Although, like with anything, there are some trade-offs to consider.
Of course.
Ion Torrent might not always have the same level of accuracy as some of the other platforms, especially when we're talking about those longer reads. It's something to keep in mind depending on the application.
Another reminder that there's always this balance, right? Speed, read length, accuracy. It's fascinating to see how these different technologies prioritize each of those factors.
For sure. And that's why it's so important to have this whole range of tools available.
Okay, moving on to our next platform, which our article has very dramatically named the Marathon Runner.
Gotta love those names.
I know, right? So we're talking about PacBio SMRT sequencing.
PacBio is definitely the endurance athlete of the group. This technology is known for generating incredibly long reads, some exceeding tens of thousands of base pairs.
Wow, that's really long.
It's like reading whole chapters of the genome at once.
Which I imagine makes it a lot easier to assemble those really complex genomes we were talking about. No more tiny puzzle pieces.
Exactly. And on top of that, PacBio also boasts really high accuracy, even with those extended reads.
It sounds almost too good to be true.
Well, as always, there's a trade-off. PacBio tends to be more expensive compared to something like Illumina or Ion Torrent. And its throughput, so the amount of data it can generate at once, tends to be a bit lower.
Okay, so you trade some speed and cost for those super long, highly accurate reads. Fascinating. And last but not least, we have our final contender, which the article has creatively named the Adventurer. Any guesses?
Adventurer. Is this Oxford Nanopore?
You got it.
That's a very fitting name, actually. Oxford Nanopore is really unique because it's so portable. It's like having a pocket-sized DNA sequencer.
Amazing. And that portability must open up so many doors for research, right? I mean, imagine the possibilities sequencing DNA in the field during outbreaks, even like in space.
Seriously, it's a game changer for so many fields. It's being used to do things like track the spread of diseases in real time, identify new species, even analyze ancient DNA.
That's incredible. I can see why they call it the Adventurer. But it can't all be sunshine and roses. There have to be some limitations, right?
Well, Oxford Nanopore sequencing, while becoming more and more affordable, it can still be quite pricey compared to some of those more established technologies. And the accuracy, while it's constantly improving, it's still not quite at the level of, say, Illumina or PacBio.
So it's all about picking the right tool for the job.
Exactly.
And, you know, regardless of which technology we're talking about, it's clear that HTS has just completely revolutionized genomics. It's like we've suddenly gone from having this blurry view to seeing the entire genetic world in high definition. It's incredible.
Right.
It's amazing how much ground HTS has covered in such a short time. And from the sounds of it, we're just getting started.
Oh, absolutely. This is just the beginning. HTS is constantly evolving, with new platforms and applications emerging all the time.
It's really incredible to think about. And our article from Biology Notes Online does a nice job of outlining some of those key applications.
Yeah, it gives you a really good sense of the sheer breadth of what HTS can do.
So let's dive into some of those applications. One area where I think HTS has really captured the public imagination is personalized medicine. It's a term we hear a lot, but what does it actually mean in the context of HTS?
Well, imagine a future where medical treatments are tailored to your unique genetic makeup. So instead of a one-size-fits-all approach, doctors can use your genetic information to predict your risk for certain diseases, choose the most effective medications, and even in some cases develop therapies that are specifically designed for you. That's the promise of personalized medicine, and HTS is making it a reality.
It sounds like something out of science fiction.
I know, right? But we're already seeing it happening in some areas of medicine, like oncology, for instance. HTS is being used to analyze the genetic makeup of tumors, which can then be used to guide treatment decisions and identify patients who might benefit from specific targeted therapies.
So we're talking about more effective treatments with fewer side effects.
Exactly. And it's not just about treatment either. HTS can also be used to identify individuals who are at increased risk of developing certain diseases. So we're talking about preventative medicine, early intervention, really empowering people to take control of their own health.
It's like having a crystal ball that gives you a glimpse into your future health. So we've talked about healthcare. What about other applications, like agriculture, for instance?
HTS is having a huge impact in agriculture as well. It's being used to develop more resilient and productive crops, for example. By analyzing the genomes of different plant varieties, researchers can identify genes that confer resistance to pests, tolerance to drought, or increase yield.
So we're talking about crops that can withstand the challenges of climate change and help us feed a growing population.
Exactly. And it's not just about improving existing crops either. HTS is also being used to unlock the genetic secrets of wild plants, which often harbor a lot of untapped potential.
It's like tapping into nature's own genetic library. And what about metagenomics? We touched on it earlier, but I'd love to hear more about how HTS is being used to study those complex microbial communities.
It's a whole other world, right? And HTS has really revolutionized how we study microbes. It allows us to analyze the DNA of entire communities of microorganisms all at once.
So instead of just looking at a single species in isolation, we can study how these microorganisms interact with each other and their environment.
Precisely. And this is giving us incredible insights into the role of microbes in everything from human health to the environment. For instance, HTS-based metagenomics is being used to study the gut microbiome, which we now know plays a crucial role in digestion, immunity, and even mental health.
It's incredible to think about all those microorganisms living inside of us, impacting our health and well-being in ways we never even imagined. And this is just a glimpse of what HTS is capable of.
And this is really just the tip of the iceberg. As HTS technologies continue to advance, we can expect to see even more groundbreaking discoveries and applications emerge in the coming years.
It's an exciting time to be working in genomics. Well, expert speaker, thank you so much for joining us today and for sharing your insights on this fascinating field.
It's been my pleasure. Always happy to talk about HTS.
And to our listeners, we hope you enjoyed this deep dive into the world of high-throughput sequencing. It's a technology that's changing the world in profound ways, and it's definitely one to keep an eye on.